Welcome back, budding biologists, to your video notes. Today, we're going to continue our discussion on biomolecules by covering proteins and nucleic acids. Let's make it make sense, shall we? By the end of this video, you should be able to identify the different ways that proteins are used in life and describe the role nucleic acids play in providing the instructions of life. All right, let's start with proteins. First, let's refresh our memory about monomers and polymers. Remember, monomers are the building blocks of polymers. So proteins are made up of amino acids, which are a monomer for proteins, and there are 20 different types of amino acids. And those amino acids, since there's 20 different types, can be combined in countless ways to create a wide variety of polypeptide chains, which are the polymer of proteins. We will be focusing heavily on amino acids and polypeptides when we cover protein synthesis in January, but for now, you just need to remember that the monomer is an amino acid and the polymer is a polypeptide. All right, proteins play a crucial role in our bodies. They're one of the fundamental biomolecules in living organisms, including us humans, and they're involved in a wide range of functions. The primary function we want to focus on is that proteins provide structural support for tissues. They're essential for maintaining the shape, strength, integrity, etc. of various body parts. So listed here, we do have some specific examples. I'm going to go through each one. Hair, for instance, is primarily made up of a protein called keratin. And keratin provides strength and structure to each strand of hair, making it durable and flexible. Keratin not only makes up hair, but also contributes to the formation of the outer layer of skin, nails, and even the epidermis, which is the skin's outermost layer. Essentially, it acts as a protective barrier for us. Fingernails are composed of a tough protein known as alpha keratin, and this protein gives nails their hardness and protects the fingertips. Muscle tissues are rich in proteins called actin and myosin, and these proteins allow muscles to contract and relax, enabling us to move and perform various physical activities. While actin is crucial for muscle contraction, it also is involved in cell processes. It allows for cell movement and cell division. And of course, the skin contains proteins like collagen and elastin. Collagen provides the skin strength and resilience, while elastin allows the skin to stretch and return to its original shape. So collagen is the most abundant protein in the human body that we know of, and probably also one of the most well known just because of the beauty industry. Keep in mind, though, that collagen does more than just keep us youthful. It also provides structural support to tendons, ligaments, bones. It helps maintain the elasticity and firmness of all of the tissues in your body. Proteins like this are just a few examples of the many types of proteins in our bodies. You do not need to study each of them in depth as much as I discussed them. Just know that each type of protein plays a specific role in maintaining the overall health and well-being of an individual. All right, in addition to providing structural support, that brings us to our other primary function, which is to help regulate biological processes. Proteins, for instance, play a crucial role in our immune system, which we are going to cover in Unit 3. But antibodies, which are a type of protein, recognize and bind to foreign invaders like bacteria or viruses, marking them for destruction in order to keep us healthy. Enzymes, which we will discuss in our next video, are specialized proteins that act as biological catalysts. They facilitate and speed up chemical reactions within our cells. Without enzymes, many essential reactions necessary for us to live would occur too slowly to sustain our metabolism. In addition, proteins are embedded in cell membranes. We'll discuss this in Unit 4, but these proteins are known as transport proteins, and they regulate the passage of molecules into and out of cells. And then our last example also with transport is hemoglobin, which is a protein found in red blood cells, and it binds to oxygen in the lungs in order to transport the oxygen to tissues throughout the body. This process ensures that all of our cells receive the oxygen that they need for energy production and survival. So, as you guys can see, proteins are incredibly versatile. They participate in various critical biological functions, and please write down at least these four. There are more, but please know at least these examples of protein functions. All right, that brings us to denaturing. Proteins rely on their shape in order to carry out the functions we just discussed. The shape is critical because it determines how a protein interacts with other molecules in order to perform its biological role. So we will discuss this further in our next video, but for now, please know that proteins can be what we're going to call denatured. The word denature refers to the process in which a protein loses its natural shape, typically due to changes in environmental conditions, such as temperature or pH. When a protein becomes denatured, it no longer functions properly, so it might slow down the process, or if it's exposed to too high of an unfavorable condition, it might become completely inactive. We will elaborate on this again in our next video, but here I have an example of cooking an egg. So when you cook an egg, the heat causes the protein in the egg whites to denature. And that's why the clear liquid turns solid as it loses its original structure. So that's why it turns white. Okay, let's shift our focus over to nucleic acids. The monomers of nucleic acids are called nucleotides. And these nucleotides combine to form the polymers, which are either going to be DNA, deoxyribonucleic acid, or RNA, ribonucleic acid. So again, nucleotides are the building blocks of nucleic acids, that's our DNA and RNA, and it's important to know that nucleotides are complex molecules with a specific structure that plays a fundamental role in storing and transmitting genetic information. We will cover this more in depth in Unit 5. Please draw this image right here, though. You need to know that nucleotides are composed of three parts: a phosphate, a sugar, and a nitrogen base. When these nucleotides come together, we form our polymers. Please also draw this. This is an example of DNA, and that is one of our polymers. So here's a single nucleotide coming together to create this segment of DNA. All right, DNA and RNA are both types of nucleic acids, but they have distinct roles and structures. The key structural difference between DNA and RNA is the type of sugar that they contain. DNA nucleotides have deoxyribose sugar, which lacks one oxygen atom, compared to ribose sugar in RNA. That's where they get their name, is from the sugar. So deoxyribose sugar in DNA, and then ribose sugar in RNA. The other major difference involves the nitrogen bases, which you're looking at here. In DNA, nucleotides form complementary base pairs through hydrogen bonding, and adenine pairs with thymine, and guanine pairs with cytosine. This specific base pairing is essential for the double-stranded structure of DNA. So as you can see here, DNA has two strands. We're going to call that a double helix. RNA has a single strand. So it's a single strand of RNA and then double for DNA. In RNA, we also replace thymine with uracil. So cytosine still binds with guanine, but now adenine binds with uracil. So this gives RNA that single-stranded structure. And then one last thing to note, and write this down, please. This one's important. The order of these nitrogen bases in DNA is what makes each individual's genetic code distinct. We'll talk more about that in just a minute. All right, that brings us to our functions. DNA is the primary molecule responsible for storing genetic information. Within the structure of DNA, the sequence of nucleotides encodes the genetic instructions that determine an organism's traits, characteristics, and functions. So it contains instructions for building proteins, which leads to our different traits. We have something called the universal genetic code, and that refers to the fact that all organisms on Earth, from bacteria to plants to animals, contain the same building blocks of life. They have the same nitrogenous bases, so adenine, thymine, cytosine, guanine, uracil for RNA. The only difference between us and a banana, or us and a pine cone, is the complexity and order of our nitrogen bases. So we're going to call that again the universal code. All right, finally, that brings us to the central dogma. Jot this image down, please, right here as I talk. The central dogma of biology is a fundamental concept that outlines the flow of genetic information within a biological system. It was first formulated by Francis Crick in 1958. You don't need to know that, just interesting fact. And it's become a cornerstone of molecular biology. Essentially, the central dogma describes the order of the steps through which genetic information is transferred and used to make proteins. We'll elaborate later, but for now, just know that DNA contains the information. That information is transcribed into RNA, which is translated into protein, and those proteins determine our traits. In this example, they're the reason our flower is purple. All right, let's wrap this up. We've covered a lot about proteins and nucleic acids. Remember, if anything isn't making sense, please find time to visit the tutoring center so we can break it down for you. Until then, I'll see you guys in class.